All right, let's get started for the afternoon session. My name is Haley. I'm another one of the student organizers, and I'll be the moderator for the afternoon. And it's my pleasure to welcome Waltram Ravelombola, who is a master's student at the University of Arkansas, and he studies association mapping for salt tolerance in cowpea using genotyping by sequencing.
It's my turn?
Yep.
Okay. Thanks, Haley. So I would like to thank Haley for this introduction, and thank you also to the sponsor of this plant symposium, the organizer and the participant. I really appreciate it. And thanks for coming back after the lunch and the poster session, because I know afternoon is a hard time, right, to focus on something. So as said, my name is Waltram, and I'm a master's student in cell and molecular biology from the University of Arkansas. And today I'll be talking about association mapping for salt tolerance in cowpea. So for the presentation of today, firstly, I'll be talking about some background and the scope of this study, why I'm doing this study, then the materials and methods, the results, and I will finish with some take-home messages. I think it's worth mentioning what cowpea is, because it is not a very popular crop such as rice, wheat, or soybean. So cowpea, it's a legume. It belongs to the family Fabaceae, and this is the scientific name. And cowpea, it's a diploid species. It has 11 haploid chromosomes. It's a self-pollinated crop. And the genome size is about 620 MB. In the United States, cowpea is usually known as black-eyed peas or southern peas. And at the bottom, we've got a nice picture of cowpea seeds from my advisor. It can have green color, black eye, or red Holstein, depending on the variety. So why does cowpea research matter? Up here we've got a map showing the top 10 biggest cities in 2025. And we can see that most of those big cities are in Asia, and it is currently the most populated continent. However, cowpea is mainly grown and consumed in Africa. But if we look in 2100, most of those biggest cities will be in Africa. So we can speculate an important demand in cowpea production. In addition, salinity is a big concern in agriculture. And as we know, salinity effects are severe on semi-arid areas, as found in Africa. And the cost related to salinity is estimated at $12 billion US. That was in 2002, and I'm sure that cost is increasing. With respect to cowpea, salinity significantly impacts germination, growth, and yield. So therefore, the objectives of this study were, first, to conduct an association mapping for cowpea salt tolerance, and then identifying afterwards some SNPs associated to cowpea salt tolerance at the seedling stage. We evaluated 155 cowpea accessions. 130 accessions come from the USDA germplasm, and 30 genotypes were from our breeding program in Arkansas. And we did some greenhouse evaluation by irrigating cowpea with either salt solution or deionized water. And I've used 6 pots. 3 pots were irrigated with salt solution, and the other 3 pots were irrigated with the ionized water. And this is how the greenhouse experiment looks like. We took data on genotypes without salt stress and data on the genotypes under salt stress. And we computed the salt tolerance index using this formula. And then we did some ANOVA and student t-tests for the mean separation. With respect to the genotyping section, we extracted the DNA from those genotypes using the CTAB protocol, and the GBS for the sequencing and SNP discovery were done by BGI in China. And we've used about 1,000 SNPs after filtering. And the population structure was inferred using the software and the delta K. It means how many subpopulation we have in your panel was captured using structure harvester. And the phylogenetic diversity tree was drawn using MEGA7. And with respect to the association mapping, I have used 4 models, 3 from Tassel, and 1 using an R package. So first result. Here we have a graph showing the distribution of the foliar leaf injury due to salinities on the x-axis, and on the y-axis we have a number of genotypes, and we can see that the visual leaf injury score was right skewed. Here we have a bar showing the percentage of genotypes which were trifoliate, and the percentage of genotypes which was untrifoliate. And trifoliate genotypes was the genotypes which were unable to develop the first trifoliate leaf after salt treatment application. And we can see that most of the genotypes were not able to develop the first trifoliate leaf. Up here we have 3 graphs showing the distributions of salt tolerance for plant height, fresh shoot biomass, and dry shoot biomass. And we can see that all 3 distributions were approximately normally distributed, which indicate that salt tolerance in cowpea could be controlled by a QTL for this population. Coming to the association mapping aspect, here we have 1, 2, 3 figures. The first one was obtained from structure harvester. And on the x-axis, we have a K value, and at K equal to 2, we have an optimal delta K. It means that we have 2 subpopulations in this cowpea panel. The subpopulation 1 in red and the other one in green. And on the right-hand side, we have the phylogenetic tree of the cowpea accession panel. And we can see that the results were pretty consistent with structure, because the subpopulation 1 was the genotypes belonging to the subpopulation were clustered together, and that's also the case for the subpopulation 2. Now we have a table showing the SNPs associated to salt tolerance at seedling stage. Here we have the traits, the SNP marker, the LOD value, which tells you whether your SNP is significantly associated with your trait of interest or not. And up here we have the R-square values, which tell you how much of the variation in the phenotype could be attributed to the SNP marker. And in this study, we've got a pretty relatively low R-square, which could indicate that we might have QTLs affecting salt tolerance in cowpea. And here we have the results for the other traits. The LOD was also relatively low because we had a relatively small population, and that's also the case for the R-square value. Conclusions. From this study, we could say that we found a substantial variability in salt tolerance index among the cowpea genotypes, and this is very important for plant breeders to have this variability among their germplasm. Oh, sorry for that. And we found some significant SNPs associated with salt tolerance, and here we have some lines which were highly associated, which were highly salt tolerance at seedling stage. So that's my presentation, and I would like to thank my advisor, Dr. Shi, the head of the Cell and Molecular Biology program, where I am, Dr. Rhodes, the head of the department, the head of the horticulture department, because CMB is an interdisciplinary program, Dr. McKay, Dr. Gibur, who helped me a lot with the stat, Dr. Wang, who helped me with designing the experiment properly, Dr. Garcia, Mr. Motes, who is a cowpea breeder, Mr. Wingfield, and all of the staffs from the horticulture department, Jessica Kivet, and all the lab members in Dr. Shi's lab, and last but not least, the UFplants17 Symposium organizers and participants. Thank you so much. If you have any questions, I'll be glad to take them.
All right. We have time for some questions.
Yes, sir.
I may have been still recovering from my lunch, but I didn't hear how long did you run the study? How old were the plants when you terminated the treatment?
I've done the treatment for 2 weeks. And after 2 weeks, some plants were completely died.
Any other questions? Oh, one more.
Yes.
Where does this project go from here?
Sorry, could you say that again?
Where does this project go from here? What are the next steps?
Oh, the next step is to, as we have a very susceptible line and very salt tolerance, so we are thinking of crossing those two contrasting parents to do a QTL analysis and to have a more precise location of the genomic region affecting salt tolerance in cowpea.
So are you planning to do mapping?
Yes, I do. Okay.
All right.
Thank you.
Let's